Thank you for the invitation and the kind introduction. I'm happy to be here and share with you some of our results. And I would like to encourage you right from the beginning to ask questions throughout my presentation. You don't need to wait until the very end. If something is unclear, I would much rather get a question whenever we are talking about it. Let's make it as interactive as we can. And as Raju said, I will share with you some of our advances on the technology front of developing single-cell proteomics, but I'm also going to share results that I'm very excited of how those technologies are starting to answer biological questions. And I want to start with that question of why did we develop single-cell proteomics to begin with? My training is as a computational biologist, biochemist, not so much proteomics. And one question as to why do single-cell proteomics is because organisms are heterogeneous, tissues are composed of different cells, but we can study heterogeneity with a variety of other methods that are cheaper and more accessible. For me, the motivation of developing single-cell proteomics has been to use it to answer more mechanistic questions of why and how biological systems work, and I'll share with you in the second part of my presentation some examples of starting to do that in a variety of different systems, including in the context of neurodegenerative diseases. That's an area where we are increasingly applying single-cell proteomics, in part due to some help from Raju with isolating single neurons from brains. So why is it important to study directly proteins? Perhaps this community already appreciates it, but when we think of what regulates protein abundance, there are two sides of the equation. One side is the production, the synthesis, and the other side is degradation. And what we have found is that in a number of contexts, especially in non-proliferative cells, the degradation is just as important or more influential for explaining protein abundance changes. So here is an example of having measured directly with a metabolic pulse experiment the rates of protein clearance in brain and how that correlates to protein abundance. And we find that the degradation side of the equation explains at least half of the overall variation in protein abundance. And in fact, what we see is that the less proliferative a tissue is, the more slowly cells grow, the more important protein degradation is. And in retrospect, this is very intuitive. When cells proliferate rapidly, they divide rapidly, proteins are mostly diluted before they're degraded because proteins tend to be stable. Once we start looking at non-proliferative tissues, at neurons that have been with me ever since I was born, then we see that degradation plays a major role. And we've also seen more recently a very surprising context where protein degradation plays a role. I will briefly mention this is a whole big area of research that we initiated, but we'll not have time to delve into that. And this has to do with the possibility that sometimes proteins are synthesized with sequences that deviate from the genetic code. We know that we can predict many of the protein sequences present in our bodies with the genetic code, but in some cases, the ribosomes incorporate a different amino acid. And what we found recently is by analyzing thousands of human tissues from very deep transcriptomic and proteomic data sets, that there are examples when the amino acid inserted in a protein is different from what is predicted by the genetic code. And in some cases, those cases that we measured, the proteins resulting from this alternate translation here shown in blue are actually much more stable. And this is one of the mechanisms by which they have increased abundance. And we have found many of those substitutions occurring in proteins associated with neurodegenerative diseases, such as TDP-43 and many others. And I will come back to this later at the single cell level. We are going to look again at the effect of protein degradation and how that contributes to protein abundance variation. And by the way, we see this mechanism of differential stability of proteoforms to be very influential for regulating proteoforms originating from alternative splicing. Alternatively, spliced proteoforms tend to have different stabilities, and that difference in stability is a major factor determining what is the abundance of the corresponding proteins. But with this brief introduction, let me delve more into single-cell proteomics, first the technology, and then the biology. About 10 years ago, the community of mass spectrometrists was generally skeptical about the possibility of quantifying proteins in single cells. And I was a junior PI who had estimated that we have sufficient copy numbers that present in single cells that, given the sensitivity of the technology, if we are clever enough to design data acquisition methods, sample preparation methods, we should be able to do it. And by now, we have transitioned from the initial stages of skepticism to a robustly developing field and technology that I think is ready to be widely adopted. And I would be happy to share with you some of the current capabilities and help you adopt it here at Dundee if you have interest in that. For me, the motivation of doing those measurements with mass spectrometry rather than some of the other more established approaches such as affinity reagents is the increased specificity of knowing exactly what you are quantifying and being able not only to measure abundance of an epitope, but being able to measure other functional properties of proteins, certainly modifications by phosphorylation, proteolytic cleavage, ubiquitination, protein synthesis and degradation. We've made a lot of good use of that, and I'll share some of it with you. So I think of mass spectrometry analysis of proteins, not just measuring protein abundance, but really measuring protein functions and connecting this to phenotypes and to biology. So at the moment, to give you a big picture overview of the technology, we are able to order a few thousand single cells per day for the analysis. Sample preparation was one of the important areas to develop so that we can prevent contaminants of going into the sensitive LC-MS instruments and maximize the efficiency of delivering proteins from single cells to instruments. We can analyze on a single instrument in a single day over 1,000 proteins. One way to distinguish the different methods is in two categories. One is label-free, when each single cell is analyzed one cell at a time. And label-free methods can analyze maybe up to 100, a bit over 100 single cells. The other way is to be able to barcode or label peptides from different single cells and analyze in parallel. And these are the methods that are far more scalable. At this point, we can analyze over 1,000 cells. So I'll show you some data from that. But they also have the capacity to continue scaling significantly. That's throughput, and the goal is to continue that scaling without compromising the quality of the data, the depth of coverage, the quantitative accuracy. The one aspect that is quite positive about these technologies compared to many other single-cell analysis, for example, RNA sequencing, is that we usually order millions of ions per single cell. With RNA sequencing, even when it's very deep, that's usually thousands of reads that are counted per cell. With mass spectrometry, that is many millions. So the counting statistics are much better. And those generally map to somewhere between a few hundreds to thousands of distinct proteins. And that number of what is the depth of coverage depends on the size of the cells. It depends on the technology being used and acquisition methods and so on. And I'll have more to say about it as we go forward. My group developed a few different generations for sample preparation. One of the first automated methods that we developed used multi-well sample prep, which is quite accessible. One can use fact sorting for that. But the most recent generation that we use now exclusively and we like quite a lot is doing the sample prep on glass slides. So we have these tiny droplets of a few nanoliters, and each droplet serves as the reaction vessel where a single cell is isolated, lysed, proteins digested, barcoded if need be, then pulled together and put into a multi-well plate that is ready to be inserted into autosampler. So this entire process is automated, uses the Cell-in-One instrument, and early this morning I just saw that you've acquired one. So you should be in a good position to implement this approach. And the positions of these droplets, so here you see the clusters. The clusters are present. You see, the clusters are present so that oftentimes we multiplex, and all of the cells that are going to be barcoded and ultimately pooled to be part of one set, part of one cluster. And the positions of these droplets are computationally deposited. So this is a very flexible method that one can use for label-free analysis, for multiplex, 9-plex, 18-plex, 35-plex, whatever the experimental design is. This method is easy to automate, and the miniaturization helps to prevent contaminants and to reduce losses. Now, one of the influential early observations as we started doing single-cell protein analysis was that from a single mammalian cell injected into a sensitive LC-MS system, we can detect tens of thousands, even hundreds of thousands of peptide-like features. So this is how peptide-like features look like, as many of you know. So we know that this is likely a peptide, but we don't know its sequence. And the fact was that we have the raw sensitivity to detect many, many, many copies for a large number of different peptides. And the challenge is to accurately determine their sequences. And my laboratory came up with two complementary ways of dealing with that challenge. And I'll put myself in that same position that the mass spec instrument is. I have a huge to-do list of lots of things that I can do very well, but I don't have the ability to do all of them. So the first approach that we developed is to use real-time instrument control to prioritize which are the peptides that we're going to analyze. But this is different from targeted workflows, where targeting peptides usually results in substantial limitation of the total number that we can analyze. In this case, by using different approach of real-time instrument control, we are able to focus the instrument time on identifiable peptides of interest. And this allows us to double the number of peptides that we can identify. For the experts, we are able to confidently assign sequences to 90% plus of all MS2 spectra that we acquire, which is unusual with shotgun approaches, in fact, not possible. We are able to focus instrument time consistently on the same peptides, which means that we have very high data completeness. And we're able to focus on, most importantly, on peptides of particular biological interest coming from proteins of interest, even if they're lowly abundant. So this is one of the approaches that is particularly suitable if one wanted to focus on post-translationally modified peptides. They can be prioritized, and by increasing accumulation times for those peptides, we can also increase sensitivity by at least 5 to 10-fold. It helps us increase the dynamic range. The other approach, conceptually different approach that we took to deal with the problem of having too many peptides to analyze, is to parallelize. And while I tend to think that I'm reasonably good at prioritizing, I'm not very good at parallelizing in my life. But it turns out that mass spec machines and computers are much better. So let me tell you a bit more about this approach that now we have followed. We have been able to analyze many samples in parallel using TMT for a long time, isobaric mass tags. But in that case, we're limited to fragmenting only one peptide at a time, and that certainly limits the sensitivity and the throughput. And also with data-independent acquisition, we can simultaneously fragment many peptides, but then typically only one sample at a time. What we wanted to do, the hypothesis to test, is that we can combine those two different ways of parallelizing so that we can get the multiplicative benefit. And the hypothesis was that we can increase the throughput contingent on preserving the depth of coverage and the quantitative accuracy, contingent on still sampling very large number of copies per single cell. And we teamed up with Vadim Dmitriev in my lab, and we developed the first implementation of this Plex DA, which we published a few years ago. And in that case, we demonstrated, even using old instruments, that we are able to increase throughput and, in fact, increase the number of proteins that are quantifiable across all samples while reducing the instrument time that you're using by a factor of 3 in that particular case, because we're using a 3-fold multiplexing and preserve quantitative accuracy. And in some cases, depending on the reagents that we use for multiplexing, there is, if they don't alter peptide retention time solution, there is the additional benefit that since the same peptide labeled with different tags from different samples is going to coilute together, so this is real data for a peptide, this is the precursor end from one sample, the first, this is the same peptide from the nth sample. In this case, the peptide is much less abundant, but this coelution can allow us to enhance sequence identification. And this has been very helpful for us. And here I show systematically that this is not a strange example, but in general, we are able to achieve this very high degree of coelution, much higher than what is possible to do with matching retention times across runs. And this high precision in retention times translates in higher confidence in sequence propagation. And this is some example of how data looks like from acquiring this method. So this is data from single cells analyzed. This is a peptide from a pancreatic adenocarcinoma cell and from a monocyte eluting, and we can quantify at 3 different time points the precursor. We can also quantify from the fragments. And from each of these scans, we get a quantitative estimate for the relative abundance of that peptide. And the degree to which these estimates are concordant gives us confidence about the quality of quantification. Now, one thing to say for these data here, you might think this comes from state-of-the-art mass spec. That's why we can do it. This comes from an old Q-Exactive from 2010. And to me, that's really illustrative for my own career and perhaps for students in the audience, that in 2015, 2016, when there was a lot of skepticism that we cannot do single-cell proteomics, we actually had instruments for over 5 years that had that capability, but our ability to do it was limited by beliefs of what's possible rather than by technological barriers. And as young, bright students, ambitious, trying to advance the field, it's always worthwhile to consider which of the limitations that you hear about are real limitations and which of the limitations you can change. You can push the boundaries on something that's limited by a belief rather than real technological barriers. So if I zoom out from the individual data points and we look at many proteins, we see that across the board, the proteins in single cells are quantified very well with a very high dynamic range of over 1,000-fold in relative quantification, having good accuracy and specificity. So these developments were noticed by Convergent Research and Schmidt Futures. There was a lot of enthusiasm and excitement around the establishment of single-cell proteomic methods, and they approached us with interest in accelerating the development of these technologies by having access to more resources, and this led to establishing Parallel Square Technology Institute, as Ralitsa said, about 2 years ago. So it's a very young organization. We have been gaining speed and compiling our team. We have both biological objectives that are focused around neurodegenerative diseases, so share some of that, and immunology. And we also have technological objectives in terms of scaling up and improving the quality of proteomic analysis in all different areas and applications, not just single-cell proteomics, but also specifically for single-cell proteomics. And the work that I'm going to share now, the technology side, comes from PTI. So we want to make these multiplexing reagents, barcodes, in such a way not only that we can analyze more samples, that's relatively easy, if that's the only metric to optimize, but we also want to increase the accuracy of quantification. We want to increase the confidence in sequence identification. We want to increase the sensitivity. We don't want to just get more data. We want to get better data. So to do that, we started generating libraries of small molecules made by combinatorial chemistry synthesis. And then we screen them. What is their impact on peptide fragmentation, confidence of identification? Here's an example of a first generation screen, a relatively small number of compound screen. So these 2 peptide sequences were labeled with different molecules, and each data point on this scatter plot corresponds to a different small molecule that we made as part of our combinatorial synthesis. And you can see that some of those result in not so good peptide identifications. Some of those result in more confident peptide identifications. So here we empirically identified this small molecule as having favorable properties for peptide fragmentation, and we synthesized it in very high quality isotopologs, isotopically encoded, so that we can use it either as a 5-plex mass tag barcoding reagent, and in that case, the different tags are offset by 4 Daltons, so that isotopic envelopes will not be overlapping, or as 9-plex, where we have 2 Dalton offsets between these tags. So let's look at why do I say that properties are favorable. Here you can see the fragmentation spectra for the same peptide label-free and labeled with PSMtags. You can see that the label-free fragmentation in this case looks quite good. We have a lot of Y ions. We don't have many B ions. Once we label the peptide, we see that we detect additional B ions that are not present for the label-free peptide. And this example is not a weird exception that I chose here. On the bottom, you can see the systematic analysis. If we look across all of the identified peptides, the orange bars indicate that the PSM tag labeled peptides systematically result in detecting many more B ions. Why is this important? It means that if you have those B ions, we can localize with confidence post-translational modifications. If you have those B ions, you can more confidently identify the sequence of immunopeptides, which normally don't produce many Y ions. They don't have a basic C-terminal amino acid, but once labeled with PSM tag, they result in much, much better spectra. And here's actually an example of peptides that are modeled for immunopeptides generated from RHC digest. We did de novo sequencing of controlled label-free and PSM tag labeled peptides, and we de novo sequenced 3 times more peptides once labeled with PSM tags. And this improvement in data quality is on top of the increased multiplexing capability. You're also generating 9 times more data once labeled with those tags. And by the way, remember the first part of my presentation from the second slide of identifying those protein sequences not predicted by the genetic code? We spent 5 years making sure they're real, and what we needed for that is peptide fragments. So generating more peptide fragments also gives us much more confidence to do de novo sequence and have confidence in protein products that are not as expected. Now, how about multiplexing and single-cell proteomics? That is a big motivation as to why we're developing these tags. Here in blue, you can see the performance of analyzing single-cell level samples at label-free analysis. So we quantify about 5,000 proteins, which is sort of comparable with the best results published for label-free analysis. And once we start labeling with PSM tag, we identify the same number of proteins per sample, but now we are analyzing 5-10 times samples in the same amount of time. So the throughput is much higher, here shown by the orange bars, and the quantitative accuracy is as good as for label-free. So here these box plots, many of you are familiar with, but for those who are not, I'll just say they come from mixed species, mixed proteomes from different species that are mixed into known ratios. And then we quantify the ratios in the samples and compare to the mixing ratios. And if the medians of the plots correspond to the mixing ratios, we can conclude that the accuracy is high, and this is what we see in this case. And more recently, we combine these tags with very fast gradients using a dual column approach with Ryan Kelly. So at the moment, we are able to analyze over 1,000 samples per day on a single instrument with the current generation of tags. And I'm actually about to show you that we can do even better than that. Because so far, I was talking about parallelizing in the mass domain, putting additional mass on the peptides, offsetting them in the mass domain. What if we can also offset them in the time domain? What if we start introducing the samples with a delay so that the active gradients of different samples are overlapping? This creates more complex data, but maybe we can interpret that data. And this schematic here essentially illustrates how this is going to look. with the isotopic and with the time domain multiplexing, a method that we call timeplex. So when we do that, we've demonstrated so far exceeding over 500 samples per day. And this is actually these data acquired on an old instrument. You don't need, I love all of the instrumentation advances that have happened in mass spectrometry. They're amazing. I love the Ultra, I love Astro. This data acquired on QExactive Explorers. We can still get really good data even with the old instruments if we use clever data acquisition and data interpretation approaches. So here you can see label-free. This is the throughput. This is the throughput once we start doing the multiplexing in time and in mass domain. And again, the goal is as we increase the amount of data, we also increase the quality of the data and quantitative accuracy. We don't want just more data points if they're noisy and crappy. We very much want high quality data here. And as we push the limits on parallelizing, I personally have challenged parallelizing myself. Instruments and computers are doing better, but they also have some challenges. One of those challenges is that there is higher probability of mass spec peak of mass peaks overlapping. So here's an example. This is label-free spectrum of two different peptides. The fragmentation spectrum mostly overlap if analyzed by DI. If these peptides are coislated, fragmented, many of the fragments are going to be the same and overlapping. And as we start decreasing the length of the gradients to gain throughput or multiplexing in different domains, we decrease the number of distinct fragments. So to cope with this challenge and to make our framework scalable, we developed a new open source software platform for dealing specifically with this challenge. I also mentioned that as we engineered PSMtags, we offset some of them by 2 Daltons. So in this case, we have engineered overlap in the isotopic envelope. So this is how the isotopic envelope looks once we multiplex with only 2 Dalton offset tags. And this is how our software models this as a linear superposition. Now, the question is, how well does this work? Mathematically, it's a very well-framed, perfectly posed question. But the question also is, are the data good enough to solve the inverse problem? Is the software scalable and good enough to do it? And here are the results. From, again, benchmarked with mixed species proteomes, you can see here the inferred precursor elution chromatograms for different mixing ratios, with the continuous lines being for the 5-plex. 9-plex is when we offset with only 2 Daltons, and you can see that there is a very good overlap between the two. And when we compare quantification with the spiked in proteomes, we see that the accuracy is very high. On the top is analysis with Diane, which is a leading search engine for data-independent acquisition data. And Diane can search only the 4 Dalton offset tags. Diane cannot cope with the 2 Dalton offset. JMod, the software that we developed, achieves the same quantitative accuracy both with 4 and 2 Dalton offset mass tags. Good. So this is what I wanted to share about the technological developments. And now I'll transition and tell you more about biological projects. And I've decided to give you a short vignettes of a few different areas of application rather than go in-depth dive in a particular project, but I'll be very happy to discuss in more details anything that you find interesting. One big idea that has always resonated within me is to interpret joint distributions among protein abundances activities, because they can help us distinguish between different models. For example, if we want to know if 2 kinases directly interact and phosphorylate each other, or the signal is mediated by another kinase, we can condition the joint distributions on the variable that we think mediates the interaction, and this can test between those different models. And because of the quantitative accuracy of protein measurements, which I'm going to show you here, compared to RNA sequencing, this analysis is much more meaningful, both conceptually and also technologically, to be done with single-cell protein data than single-cell RNA sequencing data. One area where we apply this covariation analysis is to identify regulators of protein transport. So we started with a population of macrophage cells that has natural heterogeneity. Single cells in any one population will be different. And we wanted to ask how this natural heterogeneity is going to influence protein transport in response to LPS stimulation, a very classical system. And we isolated single nuclei from macrophage cells so that we can measure protein transport from the cytoplasm to the nucleus. And this slide just shows technical benchmarks on number of proteins quantified in single nuclei. It's a bit over 1,000 proteins per nucleus. And the box plots again show results from mixed species experiments benchmarking the quantitative accuracy with the measured ratios corresponding very well to the mixing ratios. Now, when we looked at the data, we found that some proteins, such as NUP205, which is a subunit of the nucleopore complex, correlate positively with transport. So this means that macrophage cells that had higher abundance of NUP205 stimulation exhibited faster protein transport and more protein transport upon stimulation. And other proteins, such as VIM, correlate negatively, meaning that cells that had more of that protein reacted with less protein transport to the stimulation. And in fact, if we look at more of the proteins, we see that most of the subunit, actually all of the subunits of the nucleopore complex, are positively correlated to protein transport, which immediately suggests that protein transport is kinetically limited by the number of nucleopores. But it also shows interesting gradient of these correlations. And the gradient is such that the subunits that are part of the inner ring are more strongly correlated and more important for the rate of protein transport than those on the outer ring. And this is also correlated with their protein stabilities. And these data are suggesting the single cell covariation is suggesting hypotheses about differential involvement of different subunits of a protein complex in a biological function. But we also have many other proteins that are not part of the nucleopore complex that showed strong correlation that we hypothesize is likely to be regulatory for the transport. And to test whether this is just an association or has a regulatory effect, we use targeted knockdowns of these proteins. And we found that the transport measured in the targeted knockdowns corresponds very well to what we inferred from the covariation, supporting the possibility of interpreting protein covariation across single cells as potential hypotheses for regulatory mechanism. And here I'll tell you about another vignette of a recent experiment where we measured the rates of protein synthesis and degradation in single cells in live behaving mice. So we wanted to ask what are the layers at which the expression of genes is regulated, both transcriptional, translational, protein degradation in the context of a complex mammalian tissue. So to be able to do these measurements, we fed mice with food having stable isotopes. And from the same mice, we also isolated single cells for RNA sequencing. To increase the throughput of our analysis, we used again plexed A with non-isobaric mass tags. MTRAC in this case, we did not yet have PSMtags. So here you can see how the data is going to look for a particular peptide from two different single cells, cell 1 and cell 2. Each cell is going to have light and heavy precursors from that same peptide that we can measure and quantify, and this allows us to distinguish between the newly synthesized proteins and the abundance of the light proteins. And to give you some rough sense of scale, we analyzed a few thousand single cells by plexed A to measure protein synthesis and degradation, and 30,000, a bit over 30,000 with 10x genomics. In this particular case, we have data for about 2,000 gene products at both RNA and protein level. These cells are substantially smaller than some of the cells used for benchmarking, HeLa cells, and so on, but this also does not represent a limit on what we can do. The data was acquired with relatively old instruments, so if we were to do it now, the depth can be increased. So the first thing to do was simply to align the two different types of data sets, the protein and the RNA measurements, and this actually worked quite well. Here you can see examples for the relative protein and RNA levels for two different cell types. And if we look across all cell types, the correlated diagonal corresponds to the fact that relative protein and RNA levels generally agree for the same cell types, and that is a good thing to see. We expect that. But then we can ask more systematically to quantify the level of regulation for individual proteins and also to look at covariation. So let's look at the covariation at the RNA and the protein level for a pair of proteins within a cell type. So first, for the protein, these are two intermediate filaments that bind and form cytoskeletal fibrils in the cell. And you can see that the abundance of these proteins within the same cell type varies about 1,000-fold. So these are cells from the same cell type, and yet the abundance of that protein differs as much as 1,000-fold. And what's interesting is that this correlation is very strong to the other interacting partner that forms the same fibril. And this gives us confidence that this large variation that we are measuring is not just some noise in that protein, but it really reflects the formation of those fibrils. And the RNA data, even for these highly abundant transcripts, is much less correlated and much noisier. And on average, we find this to be very much the case. RNA data are far less well correlated for functionally related sets of proteins, certainly for protein complexes, and more difficult to interpret. Here's another example of differences in covariation where the cell type markers are very well correlated at the protein level, but much less well correlated at the RNA level. Now, let's look at the rate of protein synthesis and degradation. That was a primary objective in this experiment. We can directly estimate this for a few thousand proteins, the rates of synthesis and degradation. And because histones are not degraded to a significant degree, we mostly see increase in heavy histones upon cell division. And this is why when we look at the heavy to light ratio across single cells, we see this very distinct multimodal distribution where each mode corresponds to cells that have not divided. They have mostly light histones, cells that divided once, cells that divided twice. And from these data, we can estimate the average doubling time for each cell type in a mammalian tissue. And here are these estimates for average doubling times. And now we can look whether what we observed in the bulk data, that the rate of cell proliferation influences the mode of regulation determining protein abundance, whether that's true at the single cell level in the context of a mammalian tissue. And what we see here is that in the cells that grow most slowly, chondrocytes and smooth muscle cells, about half of all protein abundance variation is explained by protein degradation. And as the rate of cell growth increases, we go to immune cells, which are the fastest dividing cells. So we see that protein degradation now contributes only 15% or so. And this is because most proteins get diluted by cell division before they're degraded. And for translation, we see the opposite trend. We see that in fast-growing cells, in the immune cells, translation accounts for a very substantial amount of the total variance, and in the more slowly growing cells, translation determines less of the variance. And what's appealing to this is that we have to these observations. These are just direct estimates from the data. Is that we can also conceptually understand what is driving those differences in the relative contributions, and RNA abundance on average explains the contribution is not strongly correlated to growth rate, and on average explains about 30–40%. And I'll tell you about one more example where single-cell proteomics really were crucial to discover something that we couldn't detect at all at the RNA level. And this has to do with the initial symmetry breaking in mammalian development. Once upon a time, all of us were a zygote. And then that zygote became 2 cells and then 4 cells. And we wanted to know when those cells first become different. There is some data, phenotype data, suggesting that even in the 2-cell stage, the 2 cells are phenotypically different, but it has been very difficult to identify the molecular correlates to these differences. And people have done RNA sequencing, and there have been very few transcripts to be identified with differential abundance. But when we measured protein abundances at the 2-cell stage, we found that there are very consistent differences. They're so consistent that one sister blastomere always fell into one cluster, the other into the other cluster, and this allowed us to define those 2 clusters, alpha and beta. And as development progressed to late 2-cell stage, we see that the magnitude of these molecular differences increases. But then, of course, the question is, are these just protein differences with no functional consequences, or do they have some impact on the developmental phase of these cells? So to ask this question, in collaboration with the lab of Magda Zurnicka-Goetz, we split these 2-cell stage embryos into 2 cells. One cell we grew to a blastocyst to be able to characterize phenotypically how viable that cell is and what is the developmental fate. The other cell we analyzed with mass spectrometry to be able to determine whether it's alpha or beta. And we found that beta cells are far more likely to give rise to viable blastocysts, suggesting that the molecular differences also have functional consequences. And furthermore, we found that those differences, this gradient, is conserved between mouse and human embryos. And the molecular data that we had suggested that the origin of the first symmetry breaking is protein transport and protein degradation. Some ubiquitin ligases are hugely abundant in these embryos. They're orders of magnitude higher than they are in other normalized cell types. Now, finally, one last piece of biological data that we are very excited about and that we obtained thanks to a suggestion from Ralitz, as I said, is isolating neurons from human brains. We have been very interested and motivated to do single-cell analysis from brains, but of course it is challenging by any method, whether that's laser capture dissection or any other method to isolate individual cells, patch clamp, certainly challenging to do that at scale. But we worked on using a tissue disruptor, and this is how the cells to isolate look like. They have intact processes, they have processes, axons, dendrites, look morphologically characteristic for different cell types. We can also, doing mass spectrometry analysis on these cells, we can ask whether proteins from different compartments, from the cell membrane, endoplasmic reticulum, nucleus, cytoplasm, and so on, if their abundance corresponds to the abundance in intact brains, and we see that for the most part it does. So we think that at this point we have a protocol that allows us to isolate representative cells from single neurons. And astrocytes and microglia, all of the different types of cells. We are able to quantify a few thousand proteins in these cells. And again, this is not a limit. This is using old technology, not using PSMtags. With PSMtags, we can certainly increase these numbers. And many of the proteins that have known functions in neurodegenerative diseases are among those that we quantify, including tau, alpha-synuclein, TDP43, and many others. We also detect modified forms. We detect proteolytically cleaved proteoforms from these proteins. We detect some phosphorylations. We detect phosphorylation for tau and for many other proteins. So we are very excited to delve into these areas. Some of the preliminary results that we have show very systematic decrease in 20S proteosomes in Alzheimer's disease brains. And then now we are using the data that we have to identify which are some of the ubiquitin ligases that are also involved, because we see that many of the differences in protein abundance between controls and AD patients, Alzheimer's disease patients, are related to protein degradation rather than change in transcript abundance. So in the last few minutes, I'll talk about something that is exceptionally important to us briefly, but something that helped me a lot in the early days and that now we are very committed on sharing data and best practices for performing experiments. So at the beginning, I mentioned that when we first did single-cell proteomics, that was a time when many people were rather skeptical that this is even possible. And I was this young, unproven PI who said we can do it. What really helped is that we made our data available and our protocols available so that other people could scrutinize and evaluate for themselves, and they could repeat the experiments. And we put in a lot of effort in making our metadata and data easy to use, not just put them in a public databases, which is a requirement, and it's a good thing, but also try to make it particularly easy for other people to reuse and analyze the data. We have organized community guidelines. So this was an article that we published in Nature Methods for best practices for doing single-cell proteomics. Now that we have Parallel Square Technology Institute, we also organize workshops for sample preparation, data acquisition to be able to help other groups to get started and adopt best practices and adopt the current technology. As you start doing it, and I hope you will start doing single-cell proteomics here, there is certainly a lot of expertise and I think need for that. I would welcome you contributing to the next generation of these guidelines, things that you find useful. For example, one thing that wasn't present in these guidelines from 2 years ago was the potential of protein leakage. How do you characterize this? How do you quantify it? And this is something that we recently published and will be incorporated in the next generation of guidelines. At the end, I would love to acknowledge all the people and funding agencies that not only made the work possible, but they also made it a lot of fun. And specifically, I'll point to a few people here who have contributed to these projects. Andrew Leduc, holding the dog here, is the person who developed NPOP and who did the work of measuring protein synthesis and degradation rates. And Gray Huffman developed the prioritized approach. And let me see here, Harrison Specht led the development of PSMtags. Jason Derks developed Plex DA, first in my lab. Now he is a senior scientist at PTI. But in addition to those, Alex Petkowski worked on the embryo project. But in addition to the individual people whom I highlighted here, really all of these projects have been deeply, deeply collaborative. So the whole team has contributed and made them possible. I'll stop here and I'll be very happy to answer questions.
Questions, Sinequan. Right, the first student. I was just curious, have you included like targets as your analysis, like including like time series?
We can. So one of the ideas that I'm very fond of is to pause food labeled with different isotopic isotopes at different time points. So you can basically feed mice first with one isotope, then another, then a third, then a fourth. And then each cell will be recording a travel log in its proteome. And when we do the final destructive measurement of that single cell, we can infer during which period of time which proteins were synthesized and degraded. So I think that's a particularly good way of doing it because we can trace a single cell. Another way of doing it is simply sample cells and look at ensembles and clusters, how they change in time. But in that case, we don't have the association to know it's really this particular cell changing in time.
Great. Question back there.
Yeah. First of all, thank you for the talk. It was really exciting. I think we see applications of the technology. Single cell proteomics. So I have a couple of questions, if I may. One of them is, from what I can see, most of the analysis children bottom to up. Like I'm wondering how people are also kind of working, top to bottom, right? And I see maybe the technology doesn't give as much coverage as people like to, but it has been, increasing, the coverage has been increasing daily. So how feasible, first of all, would be just to try for, single cell proteomics, fixing up another like that. Top to bottom.
You can analyze intact proteins. There have been a few articles published in that regard from Alexander Ivanov's group, from John Yates' group, from Neil Califer. They all have used different approaches with different advantages and downsides. At the moment, there is a very significant benefit of being able to analyze intact protein. That's great. On the other hand, the downsides are that I mentioned with bottom-up approaches, we are counting many millions, tens, 20 millions of proteins per cell, copies, and we are getting highly quantitative measurements. With the top-down, you're counting many orders of magnitude, fewer, maybe low thousands. Certainly for the approach taken by Neil Califer, you're counting very few. The data are very noisy. You would not be able to do quantitation at single cell level, then you would have to work at the cluster. And again, it's just a trade-off. So there are certainly approaches working on the intact protein analysis is substantially more challenging, but there are certain questions that perhaps can be better addressed by the advantages of those approaches.
Small one, so which is related to this. So if you have proteins that are stable, but let's say the sequence are representing part of the whole result, how do we find the current algorithms that we use for identification? Because usually those algorithms work with databases, pasta files, so
yeah.
So would that have an impact, for example, on getting, bad impact on identification?
Well, that's why we haven't identified them. Essentially, sequencing proteins is incredibly hard. So there was a time when it was easier to sequence a protein than DNA, but it was still incredibly hard. That's why Sanger received his first Nobel Prize, and his work on sequencing insulin directly enabled and accelerated the treatment of diabetes. But for the most part, for the last 20, 30 years, with all the tremendous power of mass spectrometry and advantages and progress, we test hypotheses. We use those sequence databases, which are very useful. That's a powerful approach, but it's also looking through the proteome through a particular narrow perspective, a pinhole, because is it true that the genetic code that is found from in vitro synthesized RNAs translated in vitro by E. coli cell extract, which has very different tRNAs and ribosomes compared to the mammalian ribosomes, and clearly works very well and predicts proteins in a number of different species. It's a tremendous triumph and very successful model. But we are only testing what it predicts. We are generally not testing what it doesn't predict. We're not looking outside of it. And this is what we did in this particular case. We expanded the scope of hypotheses that we are testing. We looked at all of the peptides that are not identified from the database search, but that have a sequence similar to that of an identified peptide, except that somewhere it has a delta mass, change in mass. We don't yet know what that is. Many of those masses correspond to post-translational modifications. We found 100-fold or 1,000-fold more post-translational modifications with this approach. Many of those post-translational modifications are just boring artifacts that happen during sample prep. Some of those change in masses, a small fraction of those, corresponded to changes in amino acids. And then we applied 7 different filters to be confident that this is changing in amino acid, how this affects fragment intensities. Do we have fragments on the left, on the right? Can we localize it? Is the retention time consistent with the amino acid change? So it's a much more complex, much more elaborate process of discovery that is expanding the scope of what we considered, still not the full scope. We still did not do de novo sequencing. I think there are plenty of remaining abundant proteins that we have not characterized in this first work. but we have seen enough to think that this is very interesting, and this has given rise to a number of additional projects that now we have both in my lab and at PTI to look into that. And the technology that, for example, these PSMtags that I mentioned with increasing the fragment detection, this is wonderful. This is exactly what we need to be able to look at that in a less biased way. Still biased, but less biased way to expand the horizon of sequences that we can identify. We are also, I did not talk about it, but we have a dedicated method for de novo sequencing that we are developing that we hope is going to be orders of magnitude more powerful than what we can currently do, so that we can start transitioning from database matching fingerprinting to more powerful de novo sequencing that can sensitively and most importantly reliably identify sequences. Sorry, that was a long-winded answer, but
I have been keeping Mike waiting and then Jason.
Oh, right. Just on the chemistry of your PSM tags, which are very nice. It looks like I have 2 carboxylate, which is fine. So, what are you using to catalyze it?
So it has NH ester. Yeah. So we attach an NH ester to the. Yeah.
And then the other thing is in your time. Yeah.
Yeah.
Okay. And then in your time domain, how are you doing that? I assume you're not doing it with chemical characterization.
Yeah. No. So what we currently have are multiple columns that in which we load the sample with a time offset, and they're spraying simultaneously into the instrument, or they merge. It is physical, or they merge their output into a single spray. And what we have at the moment works beautifully as a proof of principle. It is not something that I would recommend you install in your lab, but we are working with a company to commercialize this and make something that is ready for robust, scalable implementation. And this, of course, can be applied to metabolites, to anything.
You want that incorporated deuterium now into
We have. We certainly.
You get always get a very predictable time shift in attention time for every deuterium.
I think the issue is with the predictability and the size of that time shift, because it is sort of predictable if you know the sequence, and even then maybe not as much. I think that's an option. Yeah. Mm-hmm. Yeah, I agree with you. It has potential. I think it also has some downsides, but yes, good suggestion.
Jason.
Thanks for the great presentation. Just maybe a couple outlook questions. So a lot of biomedical samples will be cells, they'll be FFPE sections and stuff. So
yeah.
Is there a cap there? That would be the first question. And the second is a routine assessment of proteins that are 4 orders of magnitude above what we're showing. You have P in every sample every day. Is there a filtering technique to take isolated elements into?
Yeah. So first, the FFP samples, that's an easy question. Everything that I described is applicable to those, except that FFP samples on average are a bit harder to analyze. We would have lower coverage, but there is nothing of what I described that is not applicable to those. The other one is if we have very lowly abundant proteins or very highly abundant, how do we deal with the dynamic range? What we have done here does not directly help with this problem, but it can take advantage of all of the normal approaches that we use for either immunodepletion, enrichment. Let's say you want to analyze plasma, you can first deplete or enrich, and then label with PSMtags. And this might be an approach that can make it possible to do the large-scale plasma analysis with mass spectrometry rather than affinity reagents.
Okay, I think we are slightly over time, so in the interest of time, I'm going to stop us now and thank Nicola. And remember.